welcome to Propel once again. And today we are very, very excited to have Dr. Kate Jeffrey, who is doing some very innovative and exciting work regarding Crohn's disease. She's on a leave of absence right now from as an associate professor of medicine at Harvard Medical School, along with being a principal investigator in immunology at the Massachusetts General Hospital and faculty at Harvard in immunology and virology. She is also an associate member of the Broad Institute, of MIT and Harvard. All of those she's on a leave of absence, but right now, presently, she's executive director at Moderna. Go ahead, Ildiko.
Welcome, Dr. Jeffrey. We are, as Annabelle said, so incredibly excited to speak with you today. I think because you cover a lot of fascinating topics that I think we don't hear enough about in the context of Crohn's disease. And I'm really looking forward to diving in on those topics and your research into epigenetics and the virome, and then, starting to think about how some of those dots may connect. But before we dive into it, I just want to give you an opportunity to introduce yourself. I understand you have some slides to share as well. So we look forward to hearing, from you, and then we can dive into some more question and answer type dialogue later on in this discussion.
Great. Thank you very much for having me. I think, as I just wanted to emphasize, I hope I can teach you some of the science that we've been, trying to understand these diseases. And I say these diseases, I don't think IBD is all one disease, but understanding the mechanisms that drive the development of these diseases, but also learning from, the patients and what it's like to have these diseases, and how that can motivate us scientists more. And particularly now that I'm in industry with the capabilities to sort of push some drugs forward, what would patients be comfortable taking? What would you really be willing to do in order to sort of prevent, cure, alleviate your disease? So with that, why don't I share some slides? I think you need to enable me to share, sorry.
Yes, I do.
So, I think today the goal is to really show you two facets of the work that we've done, really understanding the development of inflammatory bowel disease. And really that's sort of in two angles. So the first angle is understanding the contribution of something we call epigenetics to the disease. And epigenetics is how environmental cues really impact the gene expression in our cells. And actually, it's a combination of genetics and epigenetics, because we actually found a genetic mutation within an epigenetic enzyme that associates with Crohn's disease. And interestingly, not ulcerative colitis, so only Crohn's disease. And so understanding how those loss of function mutations change the action of this epigenetic enzyme really led us to not only understanding the sort of pathology that is resulting from this mutation, but also by understanding its mechanism of thinking about ways to therapeutically adjust those changes due to these mutations. And then the second part I'll get into is our work on the virome. And I'll get into that. That was work that I'm excited about, all of our work, but this one is I'm particularly excited about. I think this is just such a new area. I think we spent all of the pandemic, we're obsessed with one virus. But this idea that we humans happily exist amongst billions of viruses, and particularly those in our gut, and how the changes in these viruses may actually impact development of disease, or loss of the health-driving viruses may actually impact development of disease. So with that, I think I'm good to share. So, okay, you can see that?
Yes.
Yeah. Okay, great. So this is my lofty title of the way I think about complex immune diseases, but just really in this post-genomic era. So for those of you that sort of follow science in the media, I mean, you know about the sequencing of the human genome. So we know that we've sequenced all the genes in humans. And we really thought, oh, well, that's going to solve all diseases. We'll know every mutation that associates with every disease, and that'll be the answer to everything. I think that has given us a sense of, yeah, there's genetic mutations that associate with these complex immune diseases, but it also showed us that it's not the only cause, right, of these diseases. There's really this sort of large environmental and large epigenetic component to these diseases. So the way I think about these diseases, or not just me, the field, is that inflammatory bowel disease in particular is really this sort of triad where you have this genetic component either really driving the disease, and unfortunately sometimes in children, you know, that is a real driver. This early development is a largely genetic driven. And we know that genetic or genome-wide association studies have now associated more than 200 different genes that are mutated that associate with the likelihood that you will get inflammatory bowel disease. But having like one or many of those mutations doesn't necessarily guarantee that you'll develop disease symptoms. Often it has to be this combination of the environment. And now, what do I mean by environment? In the context of immunology, we really mean those microorganisms that live within our gut. So the bacteria and now viruses, which I'll get to, and how they are shaped by the things that we eat. So things like low fiber, for example, in the Western diet now has led to changes in the microbiome that lead to increased incidence of disease. But this grey circle here is a really key question. And can we find, you know, a single environmental trigger? Probably unlikely. It's probably going to be a range of different triggers, but having that genetic susceptibility is going to really make you more susceptible to developing disease in that context. And then epigenetics is really that bridge between those two. So how changes in our epigenome in sort of response to the environment can change the genes that are expressed. So as I mentioned, genetics is a component of this disease. But also we have this large environmental component, and I'll get into the sort of both angles that we've been looking at. So first off is just epigenetics, and I won't go into this in great detail, but other than to say that epi is Greek for above. So when we say epigenetics, it's really anything that's outside the genetic sequence of your DNA strand that controls the gene expression in your cells. And so the best way to explain that is that you have the exact same stretch of DNA in every cell type, but why is it that your liver cells have such a different gene expression program to your skin cells, to the cells in your gut? And really that is all due to epigenetics. And epigenetics really is actually a large job of it is just to keep a large component of that DNA silenced. You obviously don't want every possible gene expressed in every cell. You actually want a very specific cell type, specific program. And so epigenetics really keeps the DNA silenced. And that's just done by modifications on these things called histones that the DNA is wrapped around, or actually modifications of the DNA directly. And these modifications are driven by a collection of enzymes called writers that place these modifications, erasers that remove them, or this subfamily known as the readers, which dock to these modifications around the histones. And so the readers is really what we were focusing on, because it's really that molecular link between histone modifications and DNA modifications and gene expression. I will say that there's many enzymes that place this, and there's more than 400. And the exciting part is that many of these can actually be druggable. And so the idea is, can we reset the epigenome through drugging some of these targets? And work by myself and others had shown that, yes, indeed, small molecules can fit into these epigenetic readers and actually be used as drugs. I've realized that this is probably a lot of information, so let me just keep going. And just to say that we then, with this idea that these epigenetic readers could be a drug target, we started becoming interested in whether epigenetic readers had a designated role in the immune system first, And second, whether there was any disease association with changes in some of these epigenetic readers. And that's where we came across this protein called SP140. And so SP140 is one of these epigenetic readers in that it recognizes modified histones of which the DNA is wrapped around, and somehow, through mechanisms we didn't know, controls genes that are turned on or off in your immune cells. And that in turn just essentially regulates the function of those immune cells. And so SP140 we found actually had a very restricted expression profile to the immune system. So it was only found in cells of the immune system that we call the white blood cells. So T cells, which of course now during the pandemic, everyone's an immunologist. So you've all heard about T cells and B cells that are so important for the vaccine responses, NK cells and monocytes. So these are innate and adaptive immune cells. But what's interesting is SP140 is only expressed in the mature versions of those. It's not present in any of the stem cells. And actually, we became interested because it's actually very preferentially expressed in these inflamed human macrophages. So these are these polarized macrophages that are really responsible for that inflammatory response. They pump out a bunch of these pro-inflammatory cytokines that leads to a lot of the pain in the gut, in particular, in the case of inflammatory bowel disease. And so when we started looking into this protein further, it actually was clear that there were genetic mutations within this epigenetic reader that significantly associate with the susceptibility of Crohn's disease, and not ulcerative colitis, only Crohn's disease. So that is an interesting finding because there's actually many of those genes overlap between ulcerative colitis and Crohn's disease, but actually there are some subsets of genes that only associate with Crohn's. And so that might give us some insight into try and understand some mechanistic differences between these diseases. So this is just the genetic locus of SP140. And all I'm highlighting here is where the mutations have been found. And so these are just two mutations, which is a T to a C or a C to a T in this case. And just to cut a long story short, we sequenced peripheral blood immune cells from healthy individuals initially and looked at whether they were carrying these mutations. And this is 650 patients here. So you can see about 30% of people carry one allele or one type of these mutations, and then actually about 3%, so it's not super common, 3% of people unfortunately carry or are homozygous for these mutations. And so we found that when they carry those mutations, they actually start to lose expression of this epigenetic reader. And actually you can see here, this is the RNA and this is the protein. So these patients actually lose expression of this particular epigenetic reader. So this is a loss of function mutation. So what does that mean? Why does this loss of function mutation associate with inflammatory bowel disease? And I should say as well as multiple sclerosis. So it turns out that SP140 belongs to this large family of these epigenetic readers. And they're called SP because they're speckled protein. They look like a speck in the nucleus when you look at them down a microscope. And SP110, actually, its deficiency results in a very severe infant syndrome called VODI, or veno-occlusive disease immunodeficiency. And unfortunately, these babies die from 6 months of age, where they essentially lack B cells, one of the important white blood cells, just through a deficiency of this particular SP110. But SP140 is related, like its protein domains look very similar. But it has mutations that associate more with these complex immune diseases, so such as multiple sclerosis, Crohn's disease, as well as chronic lymphocytic leukemia. And so that suggests that there's a fundamental immune function for this epigenetic reader, but also unlike SP110, like its SP140 deficiency really is only impacted when you have this environmental component, which I'll get into. And so we found that actually, just to cut a long story short, that macrophages, which are an innate immune cell, are actually, their function is completely almost lost in the absence of this epigenetic reader. Now, these macrophages normally serve as our first line of defense against pathogens that that enter us, so viruses and bacteria. But of course, as you all know, we're constantly living with microorganisms in our gut. And our immune system is constantly responding to those, and that sort of educates our immune system. The response to these microorganisms that are in our gut is a way in which our immune system is essentially shaped. And so many genes actually that are loss of function in IBD actually are loss of function of innate immunity. So you actually have this repressed innate immune response, which is actually sort of losing any benefit of your immune cells that are gaining from the healthy bacteria in your gut or viruses. And so we put SP140 in that category. So the red line here is in mouse macrophages that lack SP140. And this one here is human macrophages that lack SP140. And you can just see that those cells are hypo-responsive. And if we take cells from patients that carry those Crohn's disease associated mutations and take them out of blood, put them in a dish in the lab, and we stimulate them, you can see that those Crohn's disease patients, now this is a well-controlled experiment because here in blue, these are Crohn's disease patients that do not carry mutations with SP140. And in the red is Crohn's disease patients that do. So you can clearly see that there's a stratification in these two sets of Crohn's disease patients. The ones that unfortunately carry the SP140 mutation, their innate immune cells are just hypo-responsive, suggesting that these particular patients, there's a defect in responding to any bacteria in the gut. And so we then proved that the genetic studies were right in mice. If we knocked out SP140, this epigenetic reader, these mice had exacerbated intestinal inflammation in a model of inflammatory bowel disease. So in vivo, in mice, we really showed that, yes, it is a causative driving factor of this disease. So I won't go into some detail. This is like very detailed, but to understand that mechanistically, we think that this particular epigenetic reader is a silencing component of the genome. And it is, here, these green dots here is just to say that where SP140 occupies the genome in immune cells, it's all areas that are actually silenced, so that you're not normally seeing genes expressed from these regions. And so we think that SP140 really is an identity keeper of macrophages, and by silencing a large part of the DNA, it's only ensuring genes that are associated with macrophage function and identity are being turned on. And when you lose SP140, the macrophage kind of loses its identity and then just becomes hypo-responsive. So our latest work then was asking, well, how does it do this? I mean, I guess the exciting thing when you're working on something completely new, nothing is known, but the daunting thing is like, you know, what does this thing do? So we just took a very, sorry, this is not in preparation. I should have updated these slides. This is now published in Cell, and I'm happy to share the publication with anyone here. So it was a recent edition of Cell where we discovered that this particular epigenetic reader essentially interacts with many proteins, but it's a key driver of or interactor of these proteins called topoisomerases. And topoisomerases are something that actually unwinds the DNA. And so in your cell, you have nearly a meter of DNA that is wound up very tightly that you cannot see inside your cell. And so topoisomerases sort of nick that DNA, open it up to enable certain genes to be expressed. And so it's clear that SP140 interacts with these proteins. We could confirm that with something called co-immunoprecipitation, where we pull out SP140 and we can see what proteins are bound to it. So this gave us a sense of what this epigenetic reader normally does in healthy cells. And to say that we then just showed that actually SP140 is a repressor of these topoisomerases. And so here is just in purple, we took SP140 protein and we mixed that with topoisomerase, and actually we saw this dose-dependent repression of topoisomerase function. Now, in people that lack SP140, because SP140 is normally a repressor of topoisomerases, topoisomerase activity really starts to go up. And all these heat maps show here in purple and in yellow is that in the absence of SP140, topoisomerase all of a sudden is now bound to many, many more locations across the genome, and it has increased activity. And so what results of that is essentially the cells start to lose their identity, because you're starting to get activity of topoisomerases and all these genes that should normally be silenced. In particular, in macrophages, that in turn essentially makes those macrophages not able to respond properly to bacteria in the gut. And so this is our working model of this particular epigenetic reader. So when immune cells mature, so the white blood cells mature out of the bone marrow, SP140 is turned on. And when the protein of SP140 is made, it actually interacts with these topoisomerases, and it actually acts as a repressor of these topoisomerases to stop them turning on any genes that the cell does not need. And so then when you have a genetic loss of function mutation within SP140, those topoisomerases start acting in places they shouldn't, and you start losing immune cell identity. So that's what we showed in Crohn's disease patients. So what was amazing about doing this work at Massachusetts General Hospital is Rumnik Xavier has set up a large cohort of inflammatory bowel disease patients. So every patient that comes into MGH agrees to give important samples, from blood to feces to sometimes biopsies of the intestine. And all of those patients are genetically sequenced, so we know what mutations they are carrying. So we could get Crohn's disease patients that were carrying SP140 mutations and compare that to Crohn's disease patients that weren't. And all of this gel here shows is this band up here just shows that these cells from this Crohn's disease patient in red that carried this SP140 mutation, their topoisomerase activity was way up in those cells. And actually, here is an interesting approach of precision medicine, because now we've understood that this particular patient carries SP140 mutations, and we know that the normal function of SP140 is to repress topoisomerases, and we know that this patient now has increased topoisomerase activity, and actually it turns out that there's already drugs that can suppress topoisomerase activity, and that's that TPT, or it's called topotecan, which is essentially a small molecule that inhibits topoisomerases. So this could be a really great utility of precision medicine, right? You sequence the patient, we know what mutation they carry, and this particular patient would benefit from topoisomerase inhibition. And so this, of course, we've shown in cells, we haven't shown in people. But what we did show was that the gene expression profile of those cells was essentially reset with this topoisomerase, which is a topoisomerase 1 inhibitor, or etoposide, which is that ETO inhibitor. So we could just essentially reset the immune cell function and transcriptional program in the patients that had lost SP140. And then, of course, we wanted to show that this was functional in vivo. So we went back into mice. And now we had mice that were knocked out for this particular epigenetic reader SP140. And we administered either the topoisomerase 1 inhibitor or the topoisomerase inhibitor 2 inhibitor. And essentially, these are, you may be familiar from other talks, I'm not sure, but this model of IBD that we use in the lab is essentially an irritant to the intestine. And essentially, you're breaking down the epithelial barrier in the intestine. And then these mice begin to lose weight as a sign of the inflammation. And so we can measure weight over time. So here in red, you can see that without SP140, these mice lose much, much more weight than their controls. But what was exciting to us is that in vivo, if we administer either topoisomerase 1 or topoisomerase 2 or a combination to these mice, we could really start to rescue this weight loss. And so that was really proof of going from not knowing anything about this particular epigenetic enzyme, understanding its function through that proteomic work. We identified it to interact with topoisomerases, and then we showed that in Crohn's disease patients that carry these mutations, that activity goes up. And in proof of principle in mice, that actually you could start to rescue phenotypes by giving those inhibitors. And another crude readout we use in mice is a colon length, so shorter colon, more inflammation. So in red here, with lacking SP140, there's this increased inflammation. that we can rescue with these topoisomerase inhibitors. And just another readout is lipocalin, which actually clinically is calprotectin. Many of you may have had that measured. That here in red, more in the absence of SP140 that we can rescue with this topoisomerase inhibition. So we are very excited by these findings because I think this is the path that us scientists really like to take in terms of genetic studies. You have this unbiased genetic association with the disease, and then we track back and understand what the function of that gene or protein normally is, and then how can we understand how its mutations lead to disease, and what does that tell us about the pathology of the disease. But also importantly, well, how does that educate us about therapeutic approaches to that disease for those people with those particular mutations? And so essentially, SP140 normally is a silencer of gene expression. And then when you carry those mutations, that silencing is lost. And topoisomerase activity goes up, and you start to see a bunch of, to use my epigenetic analogy of epigenetics being the keys of the piano that are played, essentially, normally SP140 silences all the keys that macrophages don't need, and when it's lost, you get many more of those keys being turned on. And so Crohn's disease patients from MGH that had this mutation in SP140 did have increased topoisomerase activity in their cells. And that led to this dysregulated innate immune gene expression program. And essentially, the idea from our functionality work was we showed that we could really restore those immune defects by utilizing topoisomerase inhibitors that are already FDA-approved drugs, actually. So we would be really excited to move that to the next level of screening patients that carry that mutation. And trialing whether this topoisomerase inhibition is the right approach for those individuals. So that's the epigenetic side, and I'm more than happy to stop there and take some questions, and unless I can then go on to our virome work.
Sure. Let's maybe stop and we can ask a couple of questions, and then we can move on to discussions of the virome, if that's okay with you.
Yeah.
Because I think this topic in and of itself is completely fascinating, and I think also new to a lot of people, because I think exactly as you said, we all know about the genome and sequencing of the genome, but this concept of the epigenome and the idea that our bodies are acting to silence genes within cells, I think that concept is probably newer to some people.
Yes.
So when we are talking about the epigenome, and you had talked about, SP140 specifically, stepping back for a minute, do you envision us in the not too distant future, sort of like how we have, genetic sequencing and epigenetic sequencing that says, well, yes, maybe in these 2 to 3% of patients, the issue is SP140, but, there may be a whole host of other similar loss of function or other mutations in the epigenome that are potentially at play for other Crohn's disease patients. Do you envision sort of a mapping of the epigenome, within the foreseeable future that is similar to how we can pretty easily and cost effectively sequence a genome?
Yeah, I think the quick answer is yes. I think in the short period of time, so we had so many technologies enable genetic sequencing. So if you think back to when the human genome was first sequenced, it took nearly 10 years and cost billions of dollars. Today in the lab, you can sequence a whole genome for a couple of dollars. So just in that span, the technology has changed so quickly and has become much more cheap. We can much more cheaply assess the human genome. And so in the last few years in the epigenome space, technologies, again, are enabling us to sequence the epigenome in a much more, with limited materials and a much more affordable fashion. And so what I would love if we got to the point in the clinic as sequencing becomes cheaper and cheaper and computational pipelines become more and more robust, that every patient, yes, you get diagnosed by your physician that looks at the villi in the intestine and other pathology readouts that say that you have inflammatory bowel disease, But in addition to that, then we would sequence your genome, we would sequence your epigenome, and we would sequence your microbiome. And that triad picture, we can start to stratify you as having this particular type of disease, and this is the right therapy for you. I mean, that's what I don't think that is outside the realm of the possibility in the next, decade or so, as these sequencing technologies become cheaper and we understand more and more about the causative nature of the epigenome in these diseases.
Yeah, absolutely, and that is where we would like to move to. I mean, that I like you have been in the pharmaceutical space, and personalized medicine is something we've talked about for decades across multiple different therapeutic areas. I think we are impatient in the IBD community to at least move closer to that in some way. And so if the starting point for that is we start to look specifically for something like SP140, I think we absolutely welcome that, even if it's a small subset of Crohn's patients potentially, because it's a proof of concept of kind of the underlying idea of personalized medicine, and really trying to get at what is it that's driving disease within each individual. And
totally. And I think there's more and less common mutations, right? And there's a number of genes that we really have characterized very well in the way that their loss of function drives disease. So some people may have heard of NOD2, NOD2. That's probably one of the most common and major drivers of inflammatory bowel disease. But even if we just got to that level, clinically, like getting this stuff over to the clinic is what is the challenge, right? So sorry if you're seeing my calendar there. Essentially, if we could at least get to that point in the short term, which I don't think is unreasonable, these systems are already set up, at least if the clinicians know what mutations you are carrying, and we can start to tailor diseases. I know at MGH there's been work even just from understanding responsiveness to existing drugs. So if you take anti-TNF therapy, we know that there's associations. If you carry this particular mutation, you're more likely to respond to anti-TNF than if you have this mutation. So honestly, in the short term, I wish we could at least diagnose with genetic sequencing and conduct clinical trials with genetic sequencing.
That's right. And there is more work, frankly, in this space in the very early onset IBD community, where there is, it's still small, but a higher prevalence of monogenetic disease. So there is more work doing genetic sequencing, immunologic panels, things like that do, I think, actually lead the way to what we ultimately want to get to, I think, in all patients, to have that kind of understanding. Right now, I think the general, answer I get is that it's, quote, not ready for prime time. And I'd really like us to move beyond that.
Well, I just also don't understand it, because I honestly, in the science world, sequencing and analyzing that sequencing can be done cheaply and in days. It's just a matter of getting it to a clinical grade level, right? And training people in the clinical field to be able to readily do that and have robust readouts. But I agree, we need to move beyond. We're not ready for prime time. Well, what's ready for prime time? I mean, these patients would love a faster track to their treatment. And so what's stopping us from sequencing them?
Well, that's right. That's exactly right. And even in my own very layperson way, I've done it myself at home on my laptop. So I know that it can be done by people a lot more intelligent and knowledgeable in the space than I am. But I know that there's more that we could be doing than what we are. But I think the whole idea of looking at the picture the way that you've presented it, I think is really, really important. I want to ask just one more question before moving on to the virome. And that's something you talked about at the beginning, which is the environmental piece. So there's a lot of talk in the IBD community, in the patient, in the parent community about the environment. And particularly, we tend to focus on diet, although it is much broader than that. Clearly, there's likely to be other environmental factors at play as well. But diet is one of those things that we can theoretically control a little bit better. And there was a phenomenal epidemiology paper out that was I just read yesterday, very recent paper from also the team, I think at Harvard and MGH yesterday, and made the argument that something around just north of 40% of IBD can be prevented with essentially a quote healthy lifestyle. And that is in line with a lot of epidemiology coming in part from the same group and others that have shown over the years some connection to, you talked about fiber, there's evidence around more specifically fruits in Crohn's disease, vegetables more in UC, red meat consumption, maybe a little bit more directly related to UC. But the concept of, we should be eating a healthy, whole foods, high fat, low fat, high fiber diet, avoiding things like emulsifiers that impact the microbiome. I mean, all of these things I think are rational to people in the patient and parent community. And we've always sort of felt that the field was a little bit behind on sort of recommendations and implementations along those lines. So my question to you, sorry for the long-winded question, back is, how does diet affect the epigenome, and can you modulate your diet to beneficially affect epigenetic function?
That's a very big question. I think that the short answer is, when we say dietary contributions to the epigenome, that's over generations, not just someone's individual life, but possibly their parents and grandparents, right? Because all of that microbiome gets passed down. So, so when we say diet and microbiome, there's immediate impact, but there's just longer term impact. I think humans eating less fiber over the last 50 years more has really, if you look at and you're all familiar with the statistics, I think it's now the order of 100,000 new people are being diagnosed with inflammatory bowel disease just in the US alone every year. And that's not because we're more acutely aware of it and we're diagnosing it more easily. That's because that's true new cases. And that's just in the US. But if you look at a global map, it's essentially my homeland of Australia, the US, Europe, and now you start to see, as China and India become more and more industrialized, incidences increasing in those areas. So there's something about our Western industrialized diet and way of living that is contributing to these diseases. So there's no question about the diet. I'm just not sure if it's over generations or in a short period. So that's the sort of first comment. The second would be, there's no question that the diet impacts the epigenome, no question, either directly or through regulation of the microbiome. But having direct connections is not yet done. Like, I can't tell you that this diet leads to this change, right? So the work would need to be done. But it's not to say that right now we can't try to adapt some of the science that's there and start to adjust diets to have any benefit from disease. And so I think there's been very well conducted clinical trials. I know out of Melbourne in Australia, where they have done controlled diet studies in inflammatory bowel disease patients and had seen some signals. But that's a hard one. I can't say exactly precisely which part of the diet changes the epigenome, but there's no question that it does.
No, and absolutely. I mean, as we enter new fields, it's and diet is such a complex topic as it is. But I would have to imagine that this sort of, again, at a very simplistic layperson level, that if I think about whether it's a piano, or I sometimes think about epigenetics as light switches turning things on and off, I my gut, for lack of a better word, tells me that what you consume and the environment and exercise and sleep and all of these other, lifestyle factors contribute to how you're turning these things on and off.
Totally. And that's a easy, cheap, addressable now thing to try. But the cautionary would be that it's not going to be a cure-all for disease, right? So there's I think they're the easy things to try now. But I think that we will still hope that certain drugs can, on top of that, even further and hopefully eventually cure disease.
Absolutely. I mean, it's sort of the basics, and then sometimes you need to move beyond the basics because whatever the underlying pathology is just requires something right. More. And clearly we're generally in that realm when we're talking about Crohn's disease. So it was just more a general comment, because I know today with the drugs that we have, patients try to modulate their diet beneficially in some circumstances, really more as an adjunct therapy to medication.
And no, I think there's obviously no physician would think that there's harm in that. That's an easy thing to adjust.
So please, I don't want to
no, and I'm loving these conversations, but I'm so excited to get to the virome, because I think it's a whole new area. I have one
question as well. So we were talking about diet as possibly influencing the epigenetics, but how about pathogens? Do they have a direct role in this as well?
Yeah, they do. So, many bacteria actually break down the fiber that we eat into what we call short-chain fatty acids. And those short-chain fatty acids are actually direct stimulators of our immune system. And so the idea is so this is actually work done by my PhD advisor, Charles Mackay, who's now back in Australia. So Charles had done a lot of this work of how fiber or low fiber starts to impact the microbiome and then in turn impact the immune system. And so normally the presence of these good bacteria that are thriving off the high fiber are then breaking that fiber down to short-chain fatty acids, and that's a good stimulator, good sort of a signal for the immune system. And so over time, as humans are eating less and less fiber, we're losing those bacteria, these good bacteria, and you're getting less of these short-chain fatty acids sort of stimulating the immune system in a positive way. So I think that the more direct one in the short term would be absolutely high fiber diet as an anti-inflammatory. Okay, so environment. Now that's a large question, and obviously we know the epidemiology. We know, unfortunately, if you are grew up and live in a Western civilized country, whether that's the food you eat or the environment that you're in, that impacts the onset of disease. The incidence of IBD is much, much higher in those countries than other countries. And so you've all heard about the microbiome, so that are the healthy bacteria in the gut, and much work has been done in that space. And so the idea is that, yes, either through dietary impact, changes in the microbiome, those healthy bacteria impacts the immune system, but we really haven't ever thought about other microorganisms. And one area that's become very new is the virome. And I'll get into that because I'm sort of excited by this concept. And I think it's an interesting time to really switch our thinking into viruses, because obviously, we've seen the impact one virus can have on the world's population when it's highly infectious and highly damaging. But we are existing amongst many, many viruses. And in terms of microorganisms in the gut, the last 20 years of research has focused almost exclusively on the bacteria that inhabit the intestine. And that's primarily because they've been really easy to look at. Scientifically, we have sort of cheap, quick methods to sort of identify the bacteria in the gut. And getting back to technologies, really the virome only emerged as a field because of sequencing technologies. We didn't have a way to identify viruses until recently through large-scale sequencing technologies that we call metagenomics, which is where you can essentially sequence DNA or RNA in the gut or any tissue, and then just align all of those sequences to known viral genomes that have been annotated in the database. And so that's how you can identify viruses. So it's much more arduous than bacteria. But that work has revealed that there are many, many viruses in our gut. So of the order of 1 million to 100 million viral-like particles that exist per milligram of feces or intestinal tissue. So think about those numbers. That's huge, massive, huge amounts of viruses. And I just wanted to put there that many of these viruses challenge what we call Koch's postulate. So Koch's postulate is an infectious disease postulate. So he was a German physician. This idea that if a pathogen causes disease, it's only found in individuals with that disease. And you can take that pathogen and cause disease, that's the concept of the Koch's postulate. So SARS-CoV-2 clearly causes COVID, and you can, you see the phenotype every time that person gets that virus. But many of the viruses in our gut challenge that, because many of them are found in so-called, asymptomatic individuals like myself. So we see many, many viruses with, I think I don't have any disease, but there's no overt disease, and there's many, many viruses amongst us all. So what are these viruses doing there? They like the microbiome. They learn to live amongst us, have this happy equilibrium, and in turn, not only learn to live amongst us, but have they learned, is there a benefit to their presence in the gut? And that's been shown in mice. So if you take a laboratory mouse, and you actually eliminate the viruses that's in the gut through a sort of cocktail of antivirals, or you knock out receptors that sense viruses in the immune system, actually those mice get spontaneous inflammation in the gut. So if you take away viruses, healthy ones, they're clearly contributing to normal homeostasis, because when they're not there, you get inflammation. And in turn, if you stop the immune system sensing them, then you also get inflammation. And one clue that this might have direct implications for IBD is that actually one very known receptor for sensing viruses in the immune system is a receptor called MDA5. And actually, again, genome-wide association studies with IBD have shown that there is a loss of function variant of MDA5 that associates with IBD. So this subset of patients may actually have changes in their virome, or have a really virome impacted phenotype of their disease. And now, because we can sequence viruses easily, we've seen multiple virome alterations in humans with not just IBD, but type 1 diabetes, HIV, graft versus host disease, but of course Crohn's and ulcerative colitis patients. So the virome is a very new field, and the idea that these metagenomic studies have associated viruses with disease doesn't necessarily say that those changes in the virome cause disease. And so from an immunology perspective, we were very interested to move beyond the correlations and ask, do these viruses that are associated with IBD, do they actually trigger or contribute to disease phenotype? And so with that, we kind of ask a very fundamental immune question. And it's easy to give this talk now because everyone's very familiar with viruses. So SARS-CoV-2 or influenza, any virus that is damaging, our immune system sees that viral RNA and senses it through these receptors called RIG-I or MDA5. And all of that leads the immune cell to trigger this antiviral response, this innate response that really the goal of that is to shut down that virus and to either kill off the cells that those viruses infect. And so we wondered what happens to the immune response to viruses that always exist amongst us? Is this the same response that we would see to our immune system responding to SARS-CoV-2 or flu, or is it different? And it's worth noting that many, many viruses in our gut are actually what we call prokaryotic viruses. They don't infect our cells, but they actually infect the bacteria. So the microbiome have these bacteriophages that viruses live within the bacteria. And so could those viruses also interact with our human immune system? And so we asked those questions. And it's feasible that you could get exposure to the bacteriophage genome in human cells when the human cells engulf bacteria and that bacteriophages exposed. So we took a approach where we, again, being at Mass General Hospital, we had access to, unfortunately, many Crohn's disease patients, or 3/4 of Crohn's disease patients undergo surgery, removing that inflamed intestine. And so we collaborated with Rich Hoden, who's the head of colorectal surgery at MGH. And we were able to get fresh colon resections of patients coming out of surgery. And we also got the content of the ileum, the fluid out of the ileum. And we wanted to really look in the upper intestinal tract of A, what viruses were there, and B, what did they do to the immune system. So through a process of filtration from these fresh surgery resections, we isolated the viruses and we could see them. So we could see viruses down a microscope. There were plenty of viruses in the actual intestinal tissue, and we could quantify those viruses. And actually, these viruses, just for the purposes of our population, there was no contaminating bacteria. This was a pure viral population that we were isolating through this filtration. And so what we simply asked, if we take viruses from an IBD, a Crohn's disease patient, an ulcerative colitis patient, or a healthy patient, what do those collective viruses do to the immune system? First of all, what viruses are there? So we used this metagenomic sequencing where we just isolated the DNA or RNA from this colon resection, and we sequenced that. And then through a computational approach, you align those sequences to known viral sequences. And this is where this stuff gets very somewhat mind-blowing, because we saw many viruses in the gut. Many of them were eukaryotic viruses of these families indicated here, and many bacteriophage of these families indicated here. And what I want to draw your attention to first is the black grey bars. And this is unknown. Currently, we don't even have sequences that are annotated to tell us what viruses these are. So we are at the cusp of a very new field. We have maybe in the annotated viral world, I think we're estimated to be at 10 to the 31 viruses on Earth. And I think only of the 50,000 of those are actually known what their sequence is and what they are, and we've actually given them a name. So it's kind of a just fascinating field. So just to say, there's many viruses we couldn't name, but what we did see was families from this, and I will just draw your mind, here is this picornavirus family. Now, picornaviruses are a single-stranded RNA virus, and they fit into a number of categories for those who have infants in preschool and things. There's Coxsackie virus is a picornavirus. There's other families, some rhinoviruses fit into this. But what we found was actually in ulcerative colitis and Crohn's disease patients consistently, we saw this elevation in this enterovirus B subfamily of the picornavirus family. And so this for us was a very unexpected finding, that all of these IBD patients, now this is limited numbers. So I do want to say that obviously because we were looking at colon resections, not feces, we actually saw it in the tissue and not the feces readily. So these are viruses that are infected in the tissue and not really shedding into the feces. So it's not feasible often always to be looking in the tissue, but just to say every single IBD patient we looked at, we did find this in their tissue. Now, the $99 question here, is this a cause or a result of IBD, right? So is it that the presence of this virus is causing disease, or is it a result of changes in the immune system as a result of the disease? I cannot tell you that at this point in time, but I do find this very compelling, that the presence of this virus in every IBD patient we looked at could indeed be a potential causative factor for this disease. So what are these enteroviruses? They're about 7,500 base pairs in length. They're a single-stranded RNA virus, and they look like this. They form this capsid around the virus. And this capsid here is the infecting capsid, and you can see them looking like this in morphology. And what's very interesting is that this family has previously been shown to also associate with type 1 diabetes. So there is a precedence for this enterovirus family to associate with other immune diseases like type 1 diabetes. So we are very, very interested in this. And I can say that I have a position at Moderna. We will be chasing some of these viruses as potential causative factors in IBD. So I'd love to have more conversations with the patients and with you and sort of looking at the feasibility of, can we vaccinate against IBD, essentially? So we asked the question first, though, what do these collective viruses that we isolated from the intestinal tissue do to the immune cells? Well, essentially, this collective viruses, we just put them on human macrophages, that innate immune cell that I keep talking about, and ask, well, what is the response to these viruses that are in the gut? And this is just a very simplistic plot to say this is the transcriptional change that occurred in these macrophages in response to these viruses. And I just want to draw your eyes to this black circle. So these are 5 individual patients where we've taken the viruses from the gut and put them on cells. And so you can see that very consistent transcriptional response from the non-IBD virome. And here in yellow and red, you can see how the virome from UC and CD completely overlapped and was completely divergent from the healthy virome. So that IBD virome was very much triggering a very different and divergent response in these innate immune cells to the healthy virome. And I won't go into that. But what were the genes? Well, it turns out that the healthy virome, so these are plots that show the changes in gene expression in cells. And essentially everything to the right of this 0 is things that went up, and everything to the left are genes that went down. And I hope that you can see that the healthy virome really seemed to be a suppressive factor. It seemed to downregulate gene expression in a bunch of these. And actually, many of these are known kind of antiviral pro-inflammatory genes. And in contrast, you can see that the ulcerative colitis virome very much provoked an inflammatory response in these cells. And similarly, so did the Crohn's disease virome. So essentially, this was the first evidence that virome changes in IBD were not just a consequence of disease, but actually could be a causative factor of disease, because if you take those viruses, put them on immune cells, they very much provoked an inflammatory response. So this suggests to us that these viruses are really contributing on their own and probably in combination with the microbiome to disease development. And so this is very consistent amongst patients. So these are all these individual dots, individual patients. So I think we got up to 15 colon resections per group. So in black is the healthy virome, and in yellow and red is ulcerative colitis, Crohn's disease, respectively. Essentially, IL-10, IL-22, and this protein called CISH are all what we call anti-inflammatory protective cytokines in the immune system. So you can see that the healthy virome very much increased their expression, whereas the IBD viromes did not. And I won't get into that. Conversely, the IBD virome, and these are all very consistent from different IBD patients, triggered this TNF and IL-6 response. And TNF, that many of you know, is a pro-inflammatory cytokine, and there's currently a drug against TNF as a potential treatment for IBD. But these viromes from IBD patients upregulated these pro-inflammatory cytokines. And I won't get into that. So then we thought, well, if healthy virome triggers this anti-inflammatory pathway and the IBD virome triggers this pro-inflammatory pathway, could we somehow utilize the healthy virome as a treatment for IBD? So could we suppress inflammation driven in IBD by giving a healthy virome? And so this is obviously, you've probably heard about fecal transfers and microbiome transplants as potential treatment. I just want to put virome transplants in addition into your head. And so these are in cells, and so there's obviously limitations, but what we showed in black is the healthy virome triggers this much pro-inflammatory interleukin-6, but the IBD virome in red very much triggered that increase in inflammation. But if we increase the ratio of healthy viruses to the inflamed in a dose-dependent manner, we could start to suppress that response. So putting a healthy virome on top of an IBD virome could actually start to suppress that inflammation. So this suggests to us that virome transfers could be a very interesting angle for IBD treatment. And I won't get into that. So to prove that in vivo, that these viruses really triggered IBD, we took the human viruses that we took from those colon resections and we put them into mice. And so you can see the viruses, we can count the viruses here in green. And essentially we did that in mice that we depleted their murine virome and then put the human viruses in them. And I won't go into that. Sorry, that's like too much information. But just to say, spontaneously, in the mice that received the IBD virome, they had this increased expansion of these pro-inflammatory macrophages in the gut. So spontaneously, that IBD virome did trigger inflammation. And then on top of that, we triggered this DSS colitis model. And you can see, as published, that I'd mentioned at the start, if you deplete mice of their virome, they get more intestinal inflammation. So this is just length of the colon. If the shorter it is, the more inflammation. Depleting the virome in grey, inflammation. We can rescue that inflammation by putting those healthy viruses into those mice, but we cannot rescue that if you put either ulcerative colitis or Crohn's disease viruses into those mice. And then just other readouts of that stool hypercalin coming back again. Increase without viruses, we can rescue that with healthy viruses, but we can't rescue that with IBD viruses. And looking at histology, if you have any appreciation for that, here is a healthy gut, very nice villi, no infiltration of immune cells. You get rid of viruses, you start to see inflammation. You put in a healthy virome, you can rescue that inflammation. But if you have any appreciation for histology, all of these purple dots here are infiltration of immune cells and essentially pathology of the intestine. And you can just see how much worse these IBD intestines look in the presence of these IBD virus. And this is details, but just to say that our experiments were controlled because if we knock out a protein called MABs, which is a downstream protein required for that MDA5 or RIG-I sensing of viruses by the immune system, we don't see any effect. So in other words, to say that you need the immune systems to see these viruses to see any effect on the gut. And so we do think that it's a direct effect of the viruses being sensed by the immune system. So just, and I think I'll skip over that just for the interest of time. So I think I just wanted to sort of emphasize that we really did show for the first time that there are plenty of viruses in the gut. As others had published, there were changes in the virome in IBD, but we went that step further and we isolated those viruses directly from patients with inflammatory bowel disease, and we showed that indeed those viruses were a contributing factor to inflammation because those IBD viruses triggered inflammation. Now, which of those viruses is triggering inflammation is the key question. Now, a challenge in this field is, unfortunately, I'll just go back to the list of viruses. It's probably a good place to have some discussion. So we identify all these viruses, but unfortunately our limitations right now in the lab is that we can't work with a lot of these individual viruses. A lot of these viruses aren't culturable. We can't readily replicate them in a lab to work with individual viruses and understand their individual function. So I think, I would put that out there. We can identify viruses, we can understand what the collective does, but I think a limitation is understanding what each individual virus is doing. What I will reassure you is that the National Institutes of Health has deemed the virome a very, very priority thing to understand. And so there will be a large increase in funding for virome research, understanding how these viruses change. We don't understand that. I can see that question coming. Okay, what causes the change in the virome? Is it diet? Is it? What I can tell you is that you are born with these bacteriophages from your mother, much like the microbiome being passed through. But many of these other viruses are acquired through life. And so that could be that sort of environmental contribution through life that you were not born with that contributes to disease. And so I'm very excited about this finding around the enterovirus, and is something that we are really, really trying to understand right now, and really, really would love to be able to have a vaccine approach to those particularly damaging viruses. So with that, I think I can take some questions on the virome.
Thank you so much. And it is incredibly interesting and exciting. And thank you again so much for this great work. It is absolutely fascinating. I'm going to take it down to, again, sort of a basic level. And I apologize because you're sharing all of this phenomenally good science, but I'm going to step back and think about it in a simplistic way. You know, oftentimes for those of us who have kids who were diagnosed with Crohn's disease or with UC, a couple of observations that we tend to make. One is that it will often follow a viral infection. And that's an observation that we make not just as parents, but that is captured in the literature as well. So I wonder if you have any thoughts on that connection and that relevance. And I can tell you in our personal case, that's exactly what happened. It was triggered by what seemed like a routine winter virus. So that's sort of the first question related to the virome and how that may be related. And the second is the observation that parents will often make, and I'm sure patients as well, that, you know, it sort of presents initially like an infection might. Again, at a very simplistic level, you will often have a low-grade fever, you know, you sort of present with many of the same symptoms that you might present in kind of an infectious state. And although the disease may change over time, that initial presentation has people asking, and Annabelle asked it earlier, you know, is there, going back to Koch's postulates, is there a single potential pathogenic, and that might be a virus, a bacteria, mycobacteria. You talked about genes that are implicated in mycobacterial sensing, you know, or, and this will be the end of my question, or as we saw with the connection between the Epstein-Barr virus and multiple sclerosis, which I know Moderna is also very familiar with.
Yeah, I can say publicly, we have our trials are going really well in the IBD space. So I'm pushing for this for IBD space.
And that is so exciting to us. I mean, I can tell you honestly, Annabelle and I both, when we saw that literature come out in multiple sclerosis last year, it was an absolute eureka moment because it validated a lot of what people had thought for a long time, that yes, these are complex immune-mediated diseases, but maybe, just maybe, there is a viral or some kind of pathogenic trigger that is targetable.
Yeah, I think I can answer that question in two ways. I think that this Koch's postulate idea of, is there a particular virus that triggers IBD? I think I'm highlighting one here that we are working very hard to show that it is indeed a trigger or at least a contributing factor to disease phenotype. And I think there's two parts to that. Now, the idea that does a virus infection itself is, you know, this sort of observation, or that a virus infection, you know, was the sort of seen before disease onset. Now, that could be a number of things, right? Like your immune system is already primed, and it was just that non-specific virus infection that then sort of tipped things over the edge. So I think you'd have to think about that observation in two ways, whether that, particularly if it's a respiratory virus, it's probably unlikely that it really accesses the gut. Some of them do, but most of them don't. So the question is whether, you know, it's the trigger or your immune system was already primed through some other factor, and that was just like the tipping point. So yes, and and this list here is long, so many of these viruses here we would put as candidates, but I would also put bacteria as candidates too. We have certain bacteria that are certain candidates, and that's not to say that like there won't be a common pathogen amongst all, and of course there's all fungi as well. I mean, there's been some beautiful work done by Thad Stappenbach's group at Washington University in St. Louis to show a very causative fungus for Crohn's disease patients. So I think you are absolutely right in asking, is there a single pathogenic trigger, or is it's a different trigger depending, and it's like your genetic susceptibility, that trigger makes it more likely for you. And so I think the latter is probably the thing. But I'm so compelled by our data that every single patient we found had this enterovirus. Now, I cannot tell you here that I know that that's a causative factor yet. But I think I want to highlight a couple of things there. I think, you know, we've proven that vaccines are very safe, and particularly mRNA vaccines. We've never seen this many humans take something in such a short time. We've had billions of people take these vaccines at this point. So we're already at a point where we say, okay, well, vaccine's very safe. Now, okay, in this case, we identify an enterovirus and we create a vaccine against it. You know, I still think there's the risk worth taking of, even if we don't know that this is a causative factor of disease, most people would be very willing to get a vaccination for a potential pathogen that might be causing their. Now, can we eliminate the disease? Do we just suppress phenotype? I'm so excited about this program that we have initiated, and I hope that I can share more soon. But from my perspective, yeah, can we vaccinate? Can we start to get rid of some of these viruses or bacteria that we think are contributing factors to particularly IBD? And you brought up MS, but you have to be aware that that took so long for that data to finally emerge. Those studies that came out, they were tens of thousands of patients. That was a huge undertaking. And I hope we'll get there with IBD.
Absolutely. But it was that proof of concept again idea that it was possible. And actually you can use a lot of those same databases. We have large data sets in IBD. And I know actually some researchers are interested in this topic of looking at that kind of connection. But you're finding here, you know, it's hard to not be excited when you find something in every single IBD patient. But
I also I can express my excitement to you as a scientist, but I also express caution to the patients that we have not. I cannot tell you that this is a causative factor at this time.
Understood.
Yeah. So are you thinking in terms of vaccine, are you thinking therapeutic or just preventative when you talk about possibility?
Right. So that's a great question. And that's stuff that we would get to in conversation when, this gets to sort of clinical trial testing phase. Obviously, I'm in a fantastic company. We can quickly design mRNAs and we can test these things. So what would make someone take a preventative? Well, it'd be family history, right? Like if you had a family history of IBD, then could we say, okay, let's just vaccinate you anyway? And then the other question is like, would a vaccine have an effect on someone already with established disease? Is the virus the initial trigger, and then the disease becomes independent of that virus, and even if you get rid of the virus, it doesn't change disease? I don't know, but the fact that we still saw a presence of this virus in these patients does suggest it's obviously, it shows it's still there, right? So can we eliminate the virus and improve disease phenotype? So I think both would be the answer. And these are the sorts of questions that, the clinical team here at Moderna and clinical trial planning team we'd love to have those conversations. Like, should we prioritize preventative? Should we prioritize therapeutic? Take any IBD patient, screen them for an enterovirus, or anyone with family history that has a more likelihood of eventually developing to prioritize those. And then there's all other questions about pediatric trials versus adult trials, etc. So we would probably try both.
It was when you were talking about the fecal transplants with when we're looking for bacteria, and that perhaps do the a viral transplant. So that would be also a possible therapeutic, right?
Yeah. So I think some of our data showed that the healthy virome was able to suppress, right?
Right. So that
and actually, it's worth saying that some filtered feces where there's no chance that bacteria got through, that actually had protective value outside of the microbiome. So there's already some data to suggest the non-bacterial component of those fecal transfers has an impact. So the first approach is, does a healthy virome, can we do a sort of fecal transfer type approach for putting a healthy virome in? And the second approach is, can we eliminate those smoking gun viruses that we think could be, the damaging viruses? But I think, I'm excited about the vaccine approach, maybe obviously being biased in a company that's made a vaccine, but I think that I'm excited by this and we have the technology to do it. It's just a matter of getting the right clinical populations to test it in, right? So
I think it's very exciting, and it's a perfect fit
Yeah, and I, again, just to caution, I do not know if this is causative, but I still think it's worth going for at this point.
Sure. That makes sense. I have one question that actually one of our other board members asked me to ask you, which is maybe related in some way to both the virome and the epigenetics. But one of the things we notice about IBD, as with MS, for example, similar, is geographic clustering.
Yes.
So we know, first of all, that there's a north-south divide where it's much more prevalent in the north, but even within those, there are specific regions that seem to have a disproportionately, high prevalence of IBD. Do you think that's potentially either a viral issue?
I just love that question. And my gut feeling, again, to use your terminology, is that there is an infection association with that geographical concentration, but we just do not have the data right now. So I think we need to be doing these geographical sampling and really looking at viruses and bacteria in those particular geographical regions. But because we know if you take viruses, there's outbreaks of certain viruses at certain locations. And so I would love if that panned out as a reason that you get these geographical sort of bursts.
Yeah, agreed. My gut, for whatever it's worth, my gut is in the same place as yours.
And I just think we are only limited by access to samples, sequencing. It's, as I tried to allude to, it's not easy to sequence the virome, and it is a lot of, and the other challenge is that you get better virome representation in the tissue. But I think if we could figure out more accessible tissues that enabled us to more rapidly screen, that would be a huge help. We are, we're putting some efforts into that of how can we screen for the presence of these viruses outside of needing colon resections, right? So that's work happening.
Absolutely. One of the very naive questions I asked very early on during the biopsy was, can we look at, not the microbiome, but also I specifically said at the time, years ago now, the viruses that are present there, can we take a look at it and say, inflamed tissue versus non-inflamed tissue? It was kind of a very, again, simplistic question.
You were ahead of your time. It's absolutely, we should have been looking at it then. And I agree 100%. I think, the other thing about viruses is that, they have niches, right? So viruses are what we call obligate pathogens, and they need host cells to replicate. And so you'll probably find them in certain niches of the gut as well. What I wanted to say too about this particular enterovirus is it is a fecal oral route. So this is not a respiratory virus. Like you wouldn't sneeze on someone and they would get it. This is literally fecal oral. So and that's the common transmission route for these gut viruses. The one you probably know that causes a lot of gut damage is norovirus, right? So that's the sort of cruise ship, when everyone comes down on a cruise ship with sickness, that's norovirus. But a lot of these viruses we found here are fecal oral. One I want to point out in the green is this Anellovirus family. I just want to, I know this is a lot of information, but that's like that Anellovirus is another one that I'm very, very intrigued by as a potential healthy virus. But I just don't have the evidence yet. So, but that's in everyone.
So I envision vaccines, diagnostics, and a probiotic that's really a viral probiotic, if you will.
Yeah, or just virome transfers, right? So like we sort of you hit it on the head. So we need better diagnosis for a virus involvement. How do we get better ways to assess viruses quickly and easily in patients and in accessible tissue? 2, vaccinating, and 3, transferring or delivering healthy viruses.
And do we know, have you looked at whether during flares, some of these viruses tend to...
Great question. And exactly that. You need those, what we call the longitudinal studies, right? over time. This graph here in C is a snapshot in time. I can't tell you whether that preceded a flare or what, but that's the type of study. So I think the geographical one is and number 2 is the longitudinal, because if you could see just pre-flare, you would absolutely then, yep, like that is a causative virus, right? And we just need those studies to be done.
Exactly.
There's a possibility of doing oral biopsies as well, because that's, at least with my son, that's how his disease first manifested. So and then like just again going back to the sort of what we call tropism of viruses, we need to understand what cells they infect, and could be an oral biopsy enough, or do you really need further down in the gut? But yes, it's a great suggestion.
Yeah. These are
so all... I hope I've left the virome field in your head, and epigenetics as well, and particularly SP140 as a potential diagnosis with SP140 mutations, specifically maybe needing that topoisomerase precision medicine, and then viruses as a potential causative factor of IBD.
This is very exciting, and we're really, really happy that you came, and you spent so much time with us explaining your research, because as I said in the beginning, I just find it very intriguing, and I wish I were a scientist so I could understand better everything that you explain, because I know there's stuff there that's really substantial. So
I also should thank all of my team. I get to talk about it, but my team are the ones that do all the work and all the hard yards in the lab, and getting all the logistics of the samples and the sequencing and the computational analysis and all of that. So really my team has been, that is always a team effort. So
well, you can thank them from, I think I can speak for everyone that, we're really very, very grateful for this kind of work. So
no, and I'm grateful to have the time with patients, because I think that just has to continually be our motivation, right? I think, science is science, and it's very important to understand, and we will continue to understand. But I think from us of just, comprehending what it's like to have this disease, and what patients are willing to take and do, and what therapies would be suitable, and what, would you be willing to get vaccinated? Would you be willing to take a virome transfer? Would you, and these types of questions helps us think about therapeutic advancements from the science as well.
Well, if like Ildiko had mentioned earlier, we are certainly willing to do whatever we need to do to help you get.
Yeah, so no, I appreciate that. Give us some time. We were working hard behind the scenes. I hope to be able to come back again, particularly with the vaccine approaches, and we can have some further conversation.
And I think the general answer to the question, I'll speak for myself, but I also think I speak for at least some subset of the community, is that absolutely patients will be willing to, whether it's a vaccine, whether it's a virus transfer, if there is a more targeted, particularly if there is a diagnostic component, if there's a more targeted therapy where you can articulate to patients, here's what we think is a specific mechanism, a specific target that we've identified that you carry as an individual, and we think this is the way that we can address that. Given the severity of these diseases, given the quality of life impact to patients, given the side effect profile of the medications, the efficacy ceiling, the lack of durability, all of those things, I think there's a huge unmet need that a new approach or new approaches like what you've identified today absolutely can be, can work towards filling. So I think there is an interest, there is a need. And so if there's anything we can do as patients, and as caregivers, please don't hesitate to reach out, because we're happy to support anything that moves that forward.
And particularly, I think these possibilities of treatment, they're not immunosuppressant. They are sort of, it sounds to me like you're just bringing the body back to a balance where there's a balance. And that's, I think for most people, they can grab onto that and they'll like this idea much better, particularly from a parent's point of view, so.
I'm excited about this. We are working hard. This is one of the biggest motivations to giving up my academic lab and coming over to a company. So I hope that we can get some of these through soon.
Well, thank you very much.
Thank you. I'm going to
And thank
stop
you.
the
Thanks for
recording.
our conversations and your great questions.